Enzymes at Work for Energy by Paige Baker, Grand Canyon University. Today I will be covering the process of glycolysis, including the energy requiring phase, energy releasing phase, and the end products. Next, I will be discussing gluconeogenesis and the multiple sources of molecules this process uses. Then I will discuss the enzyme aldolase. This will include the isoforms, expression, and regulation of expression by HNF1 and 3, C/EPB, and DBP. In the beginning of glycolysis, energy is required to start the glucose breakdown. This may seem somewhat counterintuitive since the goal of glycolysis is energy production. However, the energy expenditure at the beginning is merely an investment rather than a frivolous use of resources. This initial investment of energy allows glycolysis to proceed to not only produce a net gain of energy in this process, but further advance the cascade known as cellular respiration for an even greater net gain of energy. The energy requiring steps I have circled in purple. These two steps require the energy and the phosphate from the breaking of the ATP molecule phosphate bond. This fuels the conversion of glucose to glucose 6-phosphate by hexokinase. Next, glucose 6-phosphate is converted to fructose 6-phosphate by phosphoglucose isomerase. Next, the second energy requiring reaction can occur to convert fructose 6-phosphate into fructose 1,6-bisphosphate via phosphofructokinase. After these two energy expenditures, the next enzyme called aldolase, circled in yellow, results in fructose 1,6-bisphosphate as a 6-carbon molecule, breaking into two 3-carbon molecules denoted dihydroxyacetone phosphate on top and glyceraldehyde 3-phosphate on bottom. We will discuss this enzyme known as aldolase in much more detail later in this presentation. The dihydroxyacetone phosphate can be readily converted to glyceraldehyde 3-phosphate by triose phosphate isomerase. The remaining reactions in this process are energy-producing reactions. At this point, thanks to the conversion by triose phosphate isomerase, the cycle possesses two glyceraldehyde 3-phosphates to move forward in the pathway. These two molecules undergo phosphorylation via the reduction of NAD+ molecules to NADH, circled in green. This is also under the influence and action of glyceraldehyde 3-phosphate dehydrogenase. This produces two molecules of 1,3-bisphosphoglycerate. This production of two NADH molecules represents the first energy produced in glycolysis. The net ATP produced from each NADH molecule is dependent on their site of energy use. For example, NADH enters the electron transport chain in the kidneys, liver, and heart as NADH without losing energy. This results in an average of 2.5 ATP per NADH molecule. On the other hand, in the brain and skeletal muscle, NADH must first be converted to FADH2 before entry into the mitochondria, resulting in an average of 1.5 ATP per FADH2 molecule. In either case, the production of NADH is a production of energy. At this point, 1,3-bisphosphoglycerate is dephosphorylated by phosphoglycerate kinase to 3-phosphoglycerate. This provides a phosphate for ADP to ATP conversion. Then, phosphoglycerate mutase reconfigures 3-phosphoglycerate to 2-phosphoglycerate. Enolase then cleaves a water molecule from this conformation to yield phosphoenolopyruvate, or PEP. The last step of glycolysis involves the dephosphorylation of PEP by pyruvate kinase. This provides a phosphate for another ATP molecule to be produced. The result of this process is a pyruvate molecule circled in orange. In fact, this means that 2 pyruvate molecules are produced thanks to the 2 molecules of glyceraldehyde 3-phosphate that entered the second half of the energy-producing part of glycolysis. This means that along with 2 pyruvates, glycolysis produces 2 NADH molecules and 4 ATP molecules per glucose molecule. As we discussed before, the NADH could represent an average of either 2.5 ATP each or 1.5 ATP each if conversion to FADH2 is required. This means the total production of ATP is either 9 or 7 ATP respectively. Due to the 2 ATP used in the energy requiring portion, the net ATP from this reaction is 7 or 5 ATP respectively. This is represented more plainly as 2 NADH and 2 ATP being the net products of glycolysis. In contrast to glycolysis as the breakdown of glucose, gluconeogenesis is the production of glucose. In fact, to reverse the process we just discussed and produce glucose from pyruvate, the cost is quite high. This reaction requires 4 ATP, 2 GTP, and 2 NADH. This costly reaction typically occurs to provide the body with glucose in instances of intense exercise or periods of fasting and starvation. Also, much like glycolysis produces 2 pyruvate molecules, 2 pyruvate molecules are required for the gluconeogenesis of glucose from pyruvate. Some similar enzymes are at play in this reaction, with a few key intermediates required that are not seen in glycolysis. First, ATP is required in the first step to make oxaloacetate from pyruvate via the pyruvate carboxylase enzyme. This means 2 ATP are required, 1 for each pyruvate. Similarly, 2 GTP are needed, 1 for each oxaloacetate, to utilize PEP carboxykinase to make PEP. We will discuss this conversion in more detail in the next slide. The next reaction is represented by 2 arrows. This is because this is a reversible reaction that is shared with glycolysis. In the gluconeogenesis process, this means PEP utilizes enolase to become 2-phosphoglycerate. Another shared glycolysis reversible reaction rearranges 2-phosphoglycerate to 3-phosphoglycerate, requiring another 2 ATP, 1 for each molecule. 3-phosphoglycerate utilizes phosphoglycerate kinase to make 1,3-bisphosphoglycerate. Next, the oxidation of 2 NADH and G3P dehydrogenase activity results in 2 molecules of 3-phosphoglyceraldehyde. At this point, one of these molecules is readily transformed into dihydroxyacetone phosphate to be used in combination with 3-phosphoglyceraldehyde as well as the enzyme aldolase to form fructose 1,6-bisphosphate. This molecule becomes fructose 6-phosphate through fructose 1,6-bisphosphatase action. Then, phosphoglucose isomerase turns the fructose 6-phosphate into glucose 6-phosphate. Finally, this is formed into glucose by glucose 6-phosphatase. In this process, many enzymes in common with glycolysis are utilized. While it is true that oxaloacetate is converted to PEP, the process does require one more detailed step. Pyruvate is either transported or formed within the mitochondria. However, when the pyruvate utilizes ATP and pyruvate carboxylase to become oxaloacetate, this molecule cannot cross the mitochondria. In order to overcome this barrier, NADH is oxidized and oxaloacetate is reduced via malate dehydrogenase into malate. This formation can cross the mitochondria to be reoxidized by the same enzyme and NAD+ to oxaloacetate again. This results in neither a gain nor loss of NADH for this entire reaction. Finally, oxaloacetate utilizes GTP and PEP carboxykinase, as we discussed previously, to form PEP for the remainder of the gluconeogenesis process. Gluconeogenesis is not solely the reverse process of glycolysis, however. Gluconeogenesis encompasses a collection of reactions that all end in glucose from other derivatives. One such derivative is lactate. The left box in this illustration represents gluconeogenesis in the liver. Lactate is taken up from the blood and oxidized by the reduction of NAD+ with lactate dehydrogenase to form pyruvate. From this point, gluconeogenesis proceeds as we discussed before. The lactate in this example is supplied from the muscle in the right box. While lactate is present in small quantities in the body, high quantities are typically associated with release from skeletal muscle during strenuous exercise. This lactate is used to further fuel the muscles because strenuous exercises tends to absorb the typical glucose stores of glycogen that make gluconeogenesis so crucial to the body. Another form of fuel for gluconeogenesis are amino acids. Pictured in more detail here, these amino acids undergo transamination to enter as Krebs cycle intermediates, pyruvate, acetyl-CoA, or acetoacetyl-CoA, as pictured. Starting on the bottom right, with the green boxes, arginine, glutamate, glutamine, histidine, and proline can enter as alpha-ketoglutarate. Moving to the left, isoleucine, methionine, threonine, and valine can enter as succinyl-CoA. Next, aspartate, phenylalanine, and tyrosine can enter as fumarate. Moving up, asparagine, aspartate can enter as oxaloacetate. And in the top green box, alanine, cysteine, glycine, serine, threonine, and tryptophan can enter as pyruvate. In the white boxes, isoleucine, leucine, tryptophan can all enter as acetyl-CoA, and leucine, lysine, phenylalanine, tryptophan, and tyrosine can all enter as acetoacetyl-CoA. As you can see, based on where these items enter the cycle, they may or may not need to complete the Krebs cycle in order to become pyruvate and be used later in gluconeogenesis. So, based on where the amino acids enter, will determine how many processes they have to undergo before they can be turned back into glucose by gluconeogenesis. Finally, glycerol is another form of fuel for gluconeogenesis. This is present in fat stores and is incorporated into the pyruvate model we already discussed about halfway through the process. Glycerol kinase phosphorylates glycerol using a phosphate from ATP to make glycerol phosphate. Then, glycerol phosphate is oxidized while NAD+ is reduced to make dihydroxyacetone phosphate. This molecule enters the gluconeogenesis cycle with only 4 remaining enzyme alterations to become glucose. With glycolysis on the left occurring mainly in the muscles and brain, and gluconeogenesis on the right occurring mainly in the liver, this illustration brings together both cycles. As you can see, this illustration shows many of the overlapping transitions of these two cycles. The red arrow represents where the malate conversion occurs to release the oxaloacetate from the mitochondria for further transformation into PEP. The purple arrow represents where lactate turned to pyruvate would enter the gluconeogenesis cycle. This is also where amino acids would enter the cycle after moving through the necessary Krebs cycle transitions. Finally, the green arrow represents where glycerol would enter the cycle after becoming dihydroxyacetone phosphate. If you're looking at this comparison, the arrows in the middle on both arrows going up and down represent the same enzymes being used in each process. Towards the top, where the arrows are more diverse, it shows the enzymes that are used that are different between the cycles. For example, hexokinase used in glycolysis versus glucose 6-phosphatase used in gluconeogenesis. One of the enzymes we have already discussed is called aldolase. Aldolase is an important enzyme in both glycolysis and gluconeogenesis. As shown in this illustration, aldolase regulates the conversion of dihydroxyacetone phosphate and glyceraldehyde 3-phosphate to and from fructose 1,6-bisphosphate. In glycolysis, this is the first step down from 1 6-carbon molecule to 2 3-carbon molecules. Likewise, this is the first step up in gluconeogenesis from 2 3-carbon molecules to 1 6-carbon molecule. Aldolase enzymes are found in all living organisms. Those found in class 1 are found in animals and higher plants. This class is further divided into type A, B, and C. Type A aldolase are located primarily in erythrocytes and muscle as well as to a lesser extent the brain. Type B aldolase are located primarily in the enterocytes, liver, and kidneys. Finally, type C aldolase are located primarily in the brain as well as the ovary and the heart. Furthermore, type A and C subgroups focus their action primarily on glycolysis, while type B subgroups exert their action in both glycolysis and gluconeogenesis. The far less studied class 2 enzymes are found in prokaryotes and lower eukaryotes. This illustration breaks up the approximate distribution of the aldolase subtypes. As you can see, each subtype is not exclusive to one region, but there's definitely more in certain areas than others, as you can tell by the graphs. For example, muscle has the majority of aldolase A, while the heart and brain have majority aldolase C. The expression of aldolase B in the liver has been shown to be regulated by at least 4 transcription factors. First, HNF1, which stands for hepatocyte nuclear factor 1, is a transactivator for aldolase B expression in the liver. This is represented by the green HNF1 and green arrow. Furthermore, C/EBP or CAAT enhancer binding protein, shown in purple, is another aldolase B transactivator specifically that works on hepatocytes. DBP, or albumin D element binding protein, shown in blue, is a transactivator as well, but works specifically in hepatotoma cells, or hepatic cancer cells, shown as HCC cells in this illustration. On the other hand, HNF3, or hepatocyte nuclear factor 3, shown in red, is a strong inhibitor of aldolase B expression in the liver. This inhibition is competitive with HNF1 and non-competitive with DBP action. Both glycolysis and gluconeogenesis are heavily regulated, enzyme-controlled reactions that are pivotal for energy production and glucose generation. Glycolysis breaks down glucose to provide the body with necessary energy in the form of ATP to fuel numerous processes and life-sustaining cycles in the body. The pyruvate produced by this can also continue on into cellular respiration for vast amounts of energy production. Furthermore, gluconeogenesis produces glucose from many various sources to maintain a site of fuel, especially when the body is undergoing strenuous exercise, fasting periods, or in dangerous instances of starvation. One of the enzymes that is important in both processes is called aldolase. I've shown where aldolase activity operates here on this illustration. Composed of class 1 and class 2, aldolase is present in all living organisms. More specifically, class 1 is broken into type A, B, and C, which has specific sites of action we discussed before. Furthermore, like all enzymes, aldolase is activated or inhibited when necessary. This is done by various transactivators and inhibitors in the form of proteins. Just like enzymes carefully regulate glycolysis and gluconeogenesis, these proteins regulate enzyme activity. Overall, all of these components must work together in order to make sure the body is responding to any incoming food, stress, and other stimulus for proper functioning as a whole.